So it's 4 minutes past the hour, but I am seeing quite a few familiar faces joining the webinar. So I'm going to start and slowly introduce Francis. And by the time I'm finished the introduction, I'm sure there'll be several more colleagues online. So if those of you that don't know me, my name's Katrina Waite, and I'm a professor of clinical pharmacology and global health, and we're dialing in from Kampala, Uganda. Now, if you've been on LinkedIn recently, you'll see that Kampala has been quite an exciting place to be over the last couple of weeks. And I see many of the names on the call. You were all in the room where it happened when we had the first ever Pharmacometrics Africa conference. So this was a hybrid conference. It was physical, but also online. And if you do feel that you had FOMO, do not fear, because the recordings will be made available. And I know I certainly want to have another listen to some of the talks, because they were so rich and there was so much learning and discussion and interaction. And as a team, we thought to ourselves whether or not we should actually postpone today's webinar, perhaps because there's been so much going on, but at the same time, we really wanted to harness the momentum of the conference and keep the conversation going. And so it's with great pleasure that I introduce Francis Williams Ojara. So Francis is a lecturer at the University of Gulu in northern Uganda, and he's also a postdoctoral pharmacokinetic modeler on the maternal and infant lactation pharmacokinetic profile. He did his PhD at the Free University in Berlin with Charlotte Kluft and her group, and he brings quite a wealth of diverse and international experience to the team. And certainly for me, I really enjoy learning from Francis. And one of the questions that clinicians like myself often bring to our modeling colleagues is to do with study design, both in terms of numbers of patients, but also the sampling time points and how we make sure that the data really, really answers the question that we're trying to ask. And that's the topic of today's conversation. So I would encourage you to, Gabe, I'm just looking at the thing. I can't see the Q&A button, but is it there? Or yes, it is there. It's just my screen because I've got it's just the screen I'm using. So try and use the Q&A button when you have questions, because that makes them easier to see distinctly. When you put things in the chat is great for social, saying hello, introducing yourselves and things. But when you put your scientific questions in the chat, they tend to sort of blend into each other. So to make it easier for chairing, please pop your questions in the Q&A, and Francis will speak for a while, then we'll hand over to yourselves and hopefully the discussion will continue. So over to you, Francis.
Yeah, thanks. Can you confirm that you hear me and see my slides valid?
I see you, your slides, and I can hear you.
Wonderful. Like you said, it's really an opportunity to harness the momentum, but also advance further discussions following a very exciting week for a lot of people, at least, that were able to attend the conference in Uganda. I've seen actually quite some experienced colleagues on the call, and to me, that actually can easily change into also a learning session on some of the things. By no means am I going to present everything on the topic, but I think there will be some really interesting additions that could come from the audience in the chat. So, like you've mentioned, Katrina, I have put together a high-level presentation on how pharmacometrics can support optimal design of trials in pharmacology, be it pharmacokinetics or pharmacodynamics, or even pharmacokinetic pharmacodynamic trials. And I try to share from the experience of studies that I've been part of, but I know there could be a lot more out there in terms of how pharmacometrics is actually often used within either the drug development pipeline or even within the clinical settings. Okay, so this high-level overview is going to be structured like outlined here. One, I'll briefly introduce mathematical models in pharmacology, briefly touching on their applications. Then I'll talk about mathematical models in drug development, and then I'll spend quite a bit of time highlighting mathematical models in optimizing the design of PK and PD studies. And this is where I'm going to emphasize some case examples from studies I've been part of and the key take-homes on this subject of how models can be used actually to optimize trials. Okay, so not to introduce something that is really new, we all, the fact that we are on this call, probably we all know what mathematical models are and we know the basics of how mathematical models are developed. So a model is a description of a system using general rules and concepts. There are a lot of models out there, and sometimes I do realize people end up confusing. People do modeling within one space with, for example, I recently got a thesis to mark, and it's something on structural modeling, which is something I feel very incompetent on. But when we talk about mathematical models, we're talking about representing the PK or the PD of a drug using a system of differential equations very often. So mathematically really representing the way the drug moves in the body across time using a system of differential equations. Now, for the benefit of absolute understanding and acknowledging that there's different types of modelers on the call, we have largely speaking 3 types of models. We have the population models. There could be population PK models, which are perhaps more common than other types. And you have population PD models, but you also have population PK PD models that attempt to link the pharmacokinetics with the pharmacodynamics. And then you have these models are the ones that you would typically refer to as empirical data-driven models. Then we have the physiologically based pharmacokinetic models, which attempt to build from bottom up, really understanding the mechanistic attributes of the body, but also the drug-related properties, and you build how the drug would move within the general circulation, but also in particular organs within the body. So they help in providing that additional mechanistic insight onto what you would expect as concentrations in different compartments within the body. So POP-PK and PBPK models are compartmental in nature. PBPK models are more granular because you're able to describe in a more refined way what is happening across the different organs and tissues. And then we have quantitative systems pharmacology models. I know a few of us are exploring this. So these really go into more detailed complexity of the biochemical processes that underline responses to drugs within the body. So broadly speaking, these are 3 types of quantitative pharmacology models that you are going to find out there. So often you have the scientists focusing on 1 or 2 of all these types of models. And it's more common that if somebody is combining skills, they're mainly doing POP-PK and PBPK. But even more commonly, you find that somebody is only focusing largely on PBPK and they don't do a lot of POP-PK. But of course, there are some advantages of combining the skills. And it's something that I guess takes a lot of hard work and years of experience. Now, I'm coming from the perspective of what we see within the Milk team. And this is how we aspire to use the different modeling approaches with a lot of bias towards PBPK and population pharmacokinetic analysis. We're building capacity in PBPK at the moment. We use it, but our scholarly attributes at the moment is that we're building more of the skills. We quite we have some good competence in population analysis. So typically, you would see that you have the 2 types of modeling, and then you really want to have clinical data to inform these 2 modeling attributes. But it's not a linear relation between having the modeling skills and really the clinical data. So you'll find that before you actually move towards collecting clinical data, you may already need input from a model. For example, you could take a PBPK model and then it guides how to best collect clinical data. And that goes a bit into the direction of designing informative, but also optimal studies. And then you could have a scenario where you actually have the clinical data, and then you take the clinical data into a population PK analysis. Remember, the clinical data is collected after being informed by PBPK, and then now the data you have, you can take it through an analysis where you do POP-PK, but it's not in one direction. You could also have a scenario where you start by having population PK model, and then this informs how to actually collect clinical data. And then you use some of the clinical data to improve the mechanistic insights on what you understand about the drug. It goes without saying that to have this collection of skills, you need a bit of diversity. You're going to sometimes have people who are more experienced and technical in PBPK and not in POP-PK. And then you need to have a lot of discussions across the different technical members of the group, but also, for example, the clinical team and, maybe other people are working on in the lab to actually get this data ready for you. And then you have a lot of crosstalk that, you're working PBPK and population analysis, and then there's a bigger picture to this. How do you get whatever information that is coming out of the studies accessible, but also reusable by other groups that will then try to advance the science? So overall, when you put PBPK and population analysis together, it tends to give you a better mechanistic understanding, and then further inform better, clinical recommendations that you get. But in the nutshell, what we're going to focus on is how these techniques can be leveraged to improve trial designs. I'm going to come from the direction of population analysis. Again, you'll find it called pharmacometrics or loosely nonlinear mixed effects modeling, because it is the field I've worked in more extensively, but I'll touch on PBPK, for example, and its utility within this topic as well. So typically, you'll have data from a population of patients, and if you take concentrations across time, it will look variable across different patients. So the idea around nonlinear mixed effects modeling is to really maximize how much information you can get from data from populations of patients. So the model itself has three main components. One is the structural model component that tells you the typical concentration time profile for the entire population. And then you have the statistical models that on the one side will explain variability between different individuals within the population. And you have also intra-individual variability that describes the way the model predicted concentrations actually vary from the actual data points that you get from the analysis. And then lastly, the last component is the covariate model, which attempts to explain why one individual varies from the other within the data set. And this integrates the information you have about patient factors, for example, gender, age, rather sex, age, organ function and the likes, but also it could capture other information, for example, clinical factors that have been collected within the study. Okay, so the pharmacometric analysis, the major attributes that make it favored over standard statistical analysis, for example, if you just did a non-compartment analysis, would be that it uses information, it maximizes the use of information, and you can integrate within the workflows different data types, whether it's sparse data or intensely collected data or data that is coming from several studies. You have the capability of, pulling this data together and then getting very informative analysis. Secondly, with the right set of assumptions, you can actually develop a model that is mechanistic and exploits information, different pieces of information about the drug, the pathophysiology. You could also add in information about prior in vitro experiments or clinical and non-clinical data. So you go ahead to actually make very strong mechanistic claims by integrating information from different sources. The platform for the models enables explanation of inter-individual variability by covariates. And one of the most powerful roles of the models is what I'm going to spend a lot of time talking about is the model applications, simulation-based exploration. And these are actually the main basis for application of the models, for example, in study design. I want to quite quickly run through the role of models within the drug development pipeline. So just as a recap, we have different phases of drug development. The preclinical phase, where you have lab and animal studies and different in vitro experiments and a set of studies that really is focusing on assessing safety and biological activity. And then you have the phase 1 studies, give or take 20 to 100 healthy volunteers, with the focus of determining the safety and the dosage of the drugs. And then you have the phase 2 studies, 100 to 300 patients typically, and to assess the efficacy and adverse effects. And then phase 3 studies, which then includes a bigger number of patients, 1,000 to 3,000 patient volunteers, to verify the efficacy claims and monitor adverse events for long-term use of the drug. And then, of course, the phase 4, which often has variable length, but here you're targeting increasing the understanding of rare adverse effects, different study groups, how the drug actually works within this group, potential for highlighting new indications and the likes. So mathematical models have been actually fit within different phases of drug development for different purposes. For example, moving from the preclinical to phase 1, you see a lot of application of the models for predicting PK based on prior information that is obtained from animal data. And then you have in phase 1, understanding variability, using pop PK to understand variability and dose proportionality, how changing the doses actually affects exposure, and from that, having a basis of how one would predict outcome based on that. And then we have in phase 2, where you can actually use population models to describe the relationship between exposure and response and be able to get the right doses. And then in phase 3, where you can have model-informed confirmatory trials and extrapolations. And of course, in phase 4, depending on the research question at hand, you could use models to do just a huge variety of work. Models to inform regulatory, discussions, dosing in special populations, new indications, and the like. So this is where you could have unlimited use of the models. Okay, so optimal design of PK and PD studies using mathematical models. Like mentioned from the start, there are questions that often have to be answered in the process of designing a study. When you look at a published protocol that talks about a certain number of patients sampled at certain time points, given drug doses at certain levels for certain regimens, the question is often, what's the basis of that study design? So it becomes very difficult to go into a study without having an informed design. So what models can actually help with is to, for example, reduce the sample size that one requires to get an informative trial. Sample size in terms of the number of patients or the number of volunteers somebody recruits. They could help in reducing the absolute number of samples that somebody takes, that a trial takes from an individual patient. And then you could have other benefits like improving existing therapies and diagnostics, through model-based evaluations, especially when somebody has PK and PD data. And then, of course, in extrapolation of trial results based on the information you have in one group. If you have a good understanding of how physiological changes across different groups might affect, for example, the exposure, you can, based on the model, to actually make these extrapolations. Now, where do model-based simulations fit within the continuum of model development? I think this is really important to note. And very often, the focus is on developing a good model. And as we've had time and again, the relevance of a model only gets highlighted when you can mention how that model can be applied. So you can look at what initially was referred to as the learn and confirm cycle, where the process of model development can almost be considered as something that is always cyclic and can go in cycles until you optimize based on the new sources of information that one is getting. So if you look at this cycle from the onset, the first part here is to really develop a model informed by available data. So that is usually the first critical step. Of course, it would be better to go through the model development with data that has been informed. The data collection process is informed so that it's informative of the model. So sometimes you have scenarios where the data collected was for different questions, but of course we go ahead and try to develop models based on that data, and that informs a lot of other processes. So model development based on available data as a first step. And then you're able to, in the process of developing the model, you're able to, estimate parameters, critical parameters, whether they are pharmacokinetics or pharmacodynamic parameters, and then even go ahead to do some kind of sensitivity analysis to assess which are the most critical, processes within the model. This can be the same for population models, or it can be the same for PBPK models. And then once you have this third part, and you could actually take this model through a series of additional steps based on whether you can get additional data. For example, you could have used data from one study and you realize you cannot adequately define all the processes because of the number of patients. And then you maybe pull from another study to optimize the data. So the process of model revision can be quite iterative in nature. But once you have a model that you think is in a good shape to be applied, you can use this model for so many other things. And that takes us to the fourth step here, where you can leverage the information of the model to actually run different simulations for different scenarios. So perform virtual clinical trials to answer so many potential research questions. And this is often where the role of models in study design fits. You have a model and you want to use it to design a new trial. You already have some information about the model. You create virtual patients, and based on those virtual patients, you can ask many what-if questions that inform the further design of a study. So with the new model, you can ask several what-if questions, and then that informs development of a new trial, and the data that comes out of the trial can go a step ahead into actually optimizing the model further and giving a more concrete understanding about the PK and the PD relations that you're interested in. So if we retreat to the kind of trials we typically run, for example, within the IDI, where we're looking at complex, difficult to study populations, pregnancy and infants included in these trials, there are quite common questions that always come across. One, of course, it has to be clear what the clinical research question is. We need to have an understanding about the drug. If it is lumefantrine, what's the chemical nature of the drug? How would we expect it to behave in plasma or in breast milk like we're interested in? How many patients will be recruited? What matrix will be sampled from the patients? What will be the sample collection schedule for whatever matrix has been decided on? So you'll find that the role of mathematical models is quite integral in answering collectively a number of these questions. The one concept I want to highlight is the concept of stochastic simulations and estimations. So this really leverages the fact that there is a model that has been developed and it has the structural component that explains the typical profiles, but also has the stochastic components that explain variability across different individuals within the population, but also the residual intra-individual variability. That's the stochastic component. So all the models, the concentrations that are observed fit within a statistical framework that has to be able to capture the different inter-individual variabilities, but also the residual variabilities. So with the model at hand, which has been developed and validated, one is able to simulate virtual trials from the model, basically simulate, depending on a predefined numbers, make simulations of data, and then estimate repetitively based on the simulated data. And these estimates, the starting point is, of course, to assume models of different nature, which also taking data of different nature. And this data could be different proposed sampling schemes. And then after the simulations, you estimate the parameters, and then you compare these parameters with the parameters that came out of the primary model, what would be considered as the true parameters. And then by repeating this several times, you're able to tell which of the study design gives you the closest estimate of the parameters with the least variability. Of course, you need to predefine what is going to be the acceptable variability you have in terms of the parameter estimates. Okay, so I'll quickly go through some of the examples of how we have applied the idea of stochastic simulation and estimation in designing trials. So under the Milk Center, we have currently we have two ongoing trials, one on first-line anti-malarial drugs, and the second trial is on first-line anti-tuberculosis drugs. And the goal of the milk malaria study is really to understand the transfer of first-line anti-malarial drugs from maternal plasma into breast milk, to use that as a basis for estimating infant exposure for infants who are fully breastfeeding at that given time. So the trials, based on the design, we have taken 30 mothers, breast lactating mothers who are also breastfeeding, and their infants. So that's the target population for the milk malaria trial. And you can find the protocol. It's published. It gives more insights onto how the study designs were undertaken. So in order to evaluate the robustness of the sampling approach, we undertook a stochastic simulation and estimation exercise. Like I said, the starting point is really to have a model for lumefantrine that has been published, and based on this model, simulate data and then do parameter estimations based on different assumed study designs and compare this with what would take us the true parameter values. So based on that study design, what we've adopted and what the study design which has been used to actually collect samples is what is shown in the picture here. It is still quite complex, but what we would appreciate is that it enables us to get samples which are informative at informative times, and the number of samples that we have here are the lowest number of samples that can lead to informative parameter estimates. So we don't want a scenario where, for example, we suggest that you have to take more than desired samples from each patient, or having to keep the patient overnight at the study site as the only approach towards being able to effectively estimate the pharmacokinetic parameters. So that is one example that I would share with you. So this shows us from the different doses, and you can see what is captured as the first dose up to the 6th dose. And it fits within the typical dosing strategy for artemether-lumefantrine, where you need to take the drug 6 times. The first 2 doses are separated by 8 hours, and the next 5 doses are separated by 12 hours. In the other trial that we're running at the moment is for first-line drug-sensitive TB. We are interested in for all the 4 anti-TB drugs, but the focus at the moment is on rifampicin, which we are actively actually trying to get the data from the lab. The lab methods are being developed. But within this protocol, we also performed the stochastic simulation and estimation approach to identify what sampling schedules would give us informative parameter estimates for the pharmacokinetics of rifampicin in plasma and in breast milk. So uniquely, with insights on the PK of the drug and the way the drug is administered, we considered the first visit that has to be within the intensive phase of TB treatment. And we see that after a dose, we would take samples at different time points, 0 to 2, 4, 6 hours, 8 hours, and 24 hours after the dose. And then we would take the second PK visit coming within the So the first PK visit was within the intensive phase of treatment, and then the second one within the follow-up phase where the patient is taking 2 drugs. And that captures nicely the PK of the drug under different scenarios. So this has helped a lot, especially when you move ahead to propose your sampling designs when you're obtaining ethics. The question is always, why this number of samples? Can you do with less samples? Wouldn't you need more samples? This helps a lot when you're submitting your protocols for ethical approval, but also inherently you think about the cost associated with bioanalysis, which is not actually very cheap. So this has become integral, and actually we've done, as a group, we've engaged other groups within the institute. And for studies, upcoming clinical trials, it's good that people appreciate that you can use, PK knowledge to design trials that are more informed. So as you decide which sampling times, you already have a bit of information coming out of what is published and known about those drugs. Now, what I've shown is a case where you are focusing largely on estimating PK parameters, but you may have a scenario where the interest is to estimate PD. So I'm going to showcase one example of a trial that showcases very nicely how modeling can be entirely integral within design and analysis of data from clinical trials. So Marcus Yoga is an oncologist based in Switzerland, and he proposed a dosing algorithm for a drug called paclitaxel, which is used in treating lung cancer. My personal sentiment to this study is that I did my PhD off this trial data. So it showcases very nicely how you can actually fit modeling in the process of designing the trial, but also after you have the trial already designed and you have data coming out of it. So what happened around 2012, he proposed already an algorithm for dosing paclitaxel, which is based on the exposure of the drug that a patient has in the first cycle of treatment. So the disease area was non-small cell lung cancer, and the drug combinations here were paclitaxel plus platinum drugs, carboplatin and cisplatin. So using prior information from a series of in vitro studies, but also in preclinical trials, which showed that the paclitaxel concentration, the time that the concentration of paclitaxel is above a threshold of 0.05 micromoles per liter was significantly associated with neutropenia in treated patients. So this was the ultimate basis for targeting the exposure metric, which is time above a concentration of 0.05 micromoles per liter. So this is actually then used as a surrogate for evaluating the pharmacodynamics in terms of neutropenic response of the drug. So what happened based on a model of paclitaxel that was developed, there were simulations, virtual trials were undertaken. So basically, patients with characteristics which represent non-small cell lung cancer patients were formed, the so-called virtual populations. And then these patients, the virtual populations, were administered treatment as it would have been done in the typical clinical setting. And then several trials were simulated based on this setting. You have the PK model for paclitaxel, you have the virtual patient populations, you have the treatment, and then you have the trial conditions, which is almost what would be the setting of a clinical trial in the standard setting. So after undertaking these trials, the modeler then asked the question of which of the different sampling designs would lead to precise estimates of the time of concentration above the threshold of 0.05. And different trial designs were selected for examination. So as we can see, design one is the most intensive trial, trial design, which has about 8 sampling points. And then across the board, you have several other sampling designs that were evaluated. The ultimate conclusion was that taking one sample between 18 and 24 hours, which is approximately sample design 2, was what was adopted for the clinical trial on evaluation of the dose algorithm. So he then went ahead to propose a dosing algorithm of this nature. The point I'm making is not that you focus on the details of the algorithm, but to then make the point that the time of concentration above 0.05 would then be measured based on one sample collected between 18 and 24 hours for the patient. That means the patient would come in after receiving paclitaxel 18 to 24 hours later. You have one sample taken from them, and that sample would be used to measure the time of exposure above 0.05 micromoles per liter, and that would be a basis for adjusting the dose that the patient receives in the subsequent cycle. So that moves beyond focusing only on the PK attributes, but really taking it a step further to use the PK exposure predicted to even influence the prospective PD of the drug. And that is another dimension that one could think about applying the population PK model. So based on this dosing algorithm, the clinical trial that was later undertaken collected a lot of useful data on the PK of paclitaxel, but also on the pharmacodynamics of the drug. And there's been a series of other population models, either PK models or PD models or PK-PD models that have come out of this work. The point I'm trying to make, as we individually design and collect data in our different trials, let's pay attention that there is a lot more information that can come out of these trials beyond the initially perceived specific aim of the trial. And let's, try to keep an open mind to really interrogating the data more based on different techniques available. The last example I want to make is a study, the main PI is Katrina on this. So this is a study that showcases how PBPK modeling, the first examples I've given is mainly focus mainly on population analysis. Now this showcases how one can actually use PBPK for improving study design. And that's a case of the derived trial, which was a dose escalation trial for patients who are treated with atazanavir and ritonavir in the presence of rifampicin that we know induces liver enzymes and could potentially reduce the exposure of atazanavir. So HIV positive patients treated with atazanavir, ritonavir combinations in Uganda were enrolled in this trial, and then they were challenged with rifampicin to then have an idea about the drug-drug interactions for the two combinations. Now, the main point here is that for accurate dose selection within the study, a PBPK model was built at the University of Liverpool, and this model was used to predict the impact of the drug-drug interaction and hence guide the dose escalation to counteract that drug-drug interaction. So this model predicted that increasing the dose of atazanavir, ritonavir from 300 milligrams once daily to 300 milligrams twice daily would achieve sufficient plasma concentrations when used together with standard rifampicin dose. You could almost imagine, in the absence of prior information, how would one have conducted this trial without really having some predictions, informed predictions from a PBPK model. Ultimately, this trial led to a complex design, as you can see here. So you have the drugs administered once daily, and then when rifampicin was introduced, the atazanavir dose was escalated to twice daily based on the PBPK model. And at all times as the escalation was taking place, you could see that the patients were on dolutegravir cover just in case of the significant alteration in the plasma levels of the antiviral drug. So this emphasizes that beyond the population analysis applications that we've seen, you could also use PBPK modeling to design optimal trials, basically retaining a representative and informative number of samples to pick, but also driving down the cost of the trial, but also enhancing patient safety at the same time. So with those 4 examples that I've highlighted, I just wanted to have a general overview of where you could actually see models being used in designing trials. And I don't need to really advocate for always trying to look for model-based information in the process of designing trials. So the take-homes really for me are, pharmacometrics, which integrates multiple different types of models for PK, PBPK, and QSP, can be used to accurately quantify variability and guide drug development. That's something that is a little bit besides the point, but it is something we need to always remember. So these models can be used to guide efficient trial designs through virtual experiments, dose selection, and optimal sampling schedules. The one approach that is very commonly used is a stochastic simulation and estimation based on population models. This can actually be used to design trials and find out the optimal sampling strategies to get representative estimate of parameters. And then model-based development and clinical utilization can improve efficiency and reduce costs and enhance safety of trials. And they will go definitely fine optimizing therapies, especially in population groups that are difficult to study. So that's going to be my last slide. And I know there will be people that can add on very richly to the discussion I've started here, but I look forward to the questions and the further follow-up discussions. Thanks a lot. Over to you, Katrina, for any questions.
Great, and thank you very much, Francis. And you kept to time beautifully. I enjoyed popping up on the screen just to celebrate again the recent conference. I can see already 2 questions in the chat, which you might be able to read in the Q&A, Francis, but I think I'll also ask Henry and Joseph if they could actually just speak those questions. So Henry, are you able to unmute yourself and explain your question?
Yes, can you hear me?
Yes, beautifully.
Yes, Henry, I can hear
you. Yes, so what I was trying to say is like for a scenario where you have used the published semi-mechanistic PKPD, and then let's say you wanted to now use this let's say part of the similar pathways to predict a response of, let's say, new molecule on the market. So how would you probably think about calibrating it and calibrating that after?
Okay, should I go at that first? So actually, anyway,
Yeah.
the scenario you mentioned could be beyond extension of the model to PD, right? For the example I showed with the rifampicin and designing a trial for breast milk exposure. You imagine a scenario where the only available model in literature was only covering plasma exposure, but you want to estimate and guide a design that includes breast milk exposure. The point is you need to use the available data to extend the model out of the scenarios that the model has been developed to exist. And there goes a lot of acknowledging that that is the best guess that one would have at that point. It might not be the true depiction, but the best guess. So you can actually do things like if there's something published about the transfer of the drug from plasma to breast milk, then you use that information to create parameters that may represent that transfer, acknowledging the pharmacokinetics of the drug and the distribution of the drug into breast milk. And then based on that, you can actually extend the simulation based on the best guess you would have. And I think you can take that into even a pharmacodynamic scenario. If you know, for example, which exposure metric for the drug is most likely associated with the pharmacodynamic effects of the drug, you could actually extend the PK model, which is typically PK, to extend it to, to capturing a bit of PD. Or if it is a PK PD model, but you want to extend it to a different scenario that is also PD, you use the best guess based on the available information to extend this. So models can be extended. The limitation is in the absence of any data, you cannot validate whether the model extension actually represents the reality. But if you do not have any prior information, rather than taking a wild guess, you can have an informed guess by extending the model. I don't know if that remotely answers what you asked.
Yes, it partly does.
Yeah, thanks.
Thank you. And that's where I think interim analysis can be such a useful thing as well, because then we can actually go back and sort of look at some of those assumptions and sort of refine our models. And that's another reason why I'm quite keen to get strengthened lab capacity on some of our sites so that we can almost do that in-house and go back and continue to learn. Because as we all know, models can be good, they're all flawed, but some are useful. So Joseph, do you want to speak to your question? Joseph or Wari? Unable to unmute. Okay. So I'll start to read out the question, but from your view, so he thanks you for your insights. In your view, what's the biggest downside to this modelling, and what are those critical aspects that it will not address? Do you see a component or indeed several components in the process that still need a lot of development? Considering the progress made and the current trend, what would you advise newer researchers to focus on? Okay, so there's actually several questions in there, but what are the biggest kind of weaknesses and flaws, the sort of weakest links in this process at the moment, do you think? And given recent progress, how fast is the field moving?
Maybe to start with the last question, I think the field of pharmacometrics or quantitative pharmacology is moving exponentially fast. There's so many new resources that are coming into play, and the use of the techniques, I think they're now quite well established, both in drug development, but also in clinical practice. So you can be rest assured that this is a field that people know the potential, and it's worthwhile really having the capacity built within our settings as soon as yesterday. Now, a model is as good as the assumptions which go into it and the data that is available to inform it. So that is always going to be a constant. When you have data and you want to develop a population model, the quality of estimates or how detailed you can estimate the processes For example, if it's pharmacokinetics of a drug, how good you can estimate the different pharmacokinetic processes depends on how much data you have. So it's always cyclic. If you don't have good enough data, you can start with the data you have to actually develop develop a model that maybe is more mechanistic, by first starting with developing that basic, the simplest model you can develop, as long as it can serve the purpose of guiding how you develop a better study to collect that data. So it's always a learn and cycle, and learn, repeat, learn cycle. So it's always cyclic. You can always improve a model, and there will never be a point where you say, this is my final best model. You will have the best model for the data you have. So that can always improve. It's not only pharmacokinetics, but also the pharmacodynamics of the processes you're trying to explain.
So there's a few more questions coming, and I'm going to quickly jump to Robert's because it kind of follows on from what you have just been saying, that what could be the role of artificial intelligence in some of these PKPD modeling designs when there's limited data?
Yeah, and when we talk about how far the field has grown, you cannot talk about that without emphasizing the role of AI, and I think that's in all fields. So there's a lot of new insights that people are getting from AI. We had a chat in the morning. When you write your model codes, could actually AI play a role in improving how smartly you write the code? How you integrate, for example, if you have a huge database and you want to know which coverage might be relevant, could you do the first initial coverage selection process? Can it be AI based before you highlight coverage that you can actually ultimately take into the model? So AI is increasingly becoming integral into the modeling frameworks. Of course, you still need to learn the basics, because at the end of it, the modeling process is guided by your understanding of pharmacology. So that needs to always be clear from the onset. It's not AI which is going to do it for you, but AI can certainly improve the efficiency with which you do modeling from different perspectives. So it is something that one would need to keep an eye on as well. Yeah.
Great. And we do plan to have a webinar on this quite soon because it's one of the very hot topics at the moment. And I think we do need some more guidance on the role, how to use it well, but its limitations. So I'm going to bring together the questions of Davies and Provia. So Davies Otieno asks, how do we determine this optimal sample size for running simulations to improve study design versus for virtual clinical trials? So is the rationale different? Our sort of study design versus the virtual trial simulations. And I think that the rationale is a bit different. And perhaps related to that, because we've got just a couple of minutes, if you can weave them together. Provia is asking, modeling seems to be very key in protocol design. Is it just a nice to have, or is it a must have?
Yes, so Katrina, the question of sample size always comes up. And when you're doing the trial simulations, you don't rely on one scenario. You test so many scenarios. And that is how you're able to choose that. If I took 30 patients and collected 6 samples from these patients, is it better than taking 20 patients and considering 6 samples from the patients? Is it better than taking 30 patients and relying on maybe 2 samples? You have to explore several scenarios. It is important to acknowledge that the scenarios are not only informed by its logistics, its convenience for the patient, its bioanalytic cost. So there are a lot of factors that go into that. So it's not a one scenario tested and then you conclude on that. There are several scenarios tested. So optimal number of samples, again, that goes back to the question. Ideally, you want, your criteria is how many samples from the simulations, how many samples will give me reasonably precise parameter estimates, because at the end of it, when you collect the data, you want to have enough information coming out of the data. So your guiding principle is, if I take a PK which is based on for technical concepts, maybe one compartmental model, how much data would I need to be able to estimate the absorption, those processes a little bit more clearly. So the sample size, of course, in statistics, the more the samples, the more precise your estimates, but you try to work within the limits of cost and convenience to the patients and bioanalytic capacity, and then this guide how you decide on the sampling strategy.
1 minute to answer
Yes.
Provia's. Is it a must have or nice to have, or does it vary?
I think it adds a lot of information. It adds the informativeness to the protocol. It is better than not having a trial simulation included within the protocol. In short, I think that's how I would respond to it.
And as things get more complex and we look at more different comorbidities, drug interactions, complex populations, I think, yeah, from the type of work we're doing, it's a must have. Maybe for other clinical trials, less so. So I know we're at the top of the hour, so I had to hurry Francis on, but that's actually the sign of a good webinar when these questions keep coming. I would like to reiterate, we shall be having one on artificial intelligence, so watch out for our advert for that. And I notice we're getting quite a regular attendance. And if you feel there's a topic that you would really like to cover, we also are very open to respond to that. So for today, I want to thank you all for your attention. Thank you for the discussion. I certainly learned something. I'm sure you did too. Thank you, Francis.
Thank you. Bye-bye, everyone.